Thank you so much for joining us for today's science talk. We're so pleased you could join us for this important session on nanoplastics in our environment, small particles with big challenges. My name is Dr. Miriam Axel, and I'm thrilled to be your host today. For those of you joining us for the first time, welcome. We're so glad you could make it. This is our weekly series that showcases the exciting research of UNU-INWEH's affiliates. I'm pleased to share that all of our previous science talks are available on our website and online at our website, unu.edu/INWEH/science-talks with an S at the end. Today's talk, like all of our others, will be recorded, and the recording will be made available after the session. We also invite you to please put your questions for our speaker in the Q&A box, and I will be sure to read them out to our speaker at the end of the session. Don't wait until the very end. Feel free to put your questions in at any point during the talk, and we will get to them at the very end. And now it's my great pleasure to introduce today's speaker, Professor Nathalie Tufenkji, is an adjunct professor in water quality and plastic pollution here at UNU-INWEH. And her research delves into particle surface interactions with a focus on safeguarding water resources, combating plastic pollution, and discovering natural antimicrobials. At UNU-INWEH, she contributes to our research and capacity building initiatives on water quality and plastic pollution, covering topics such as water and wastewater treatment, microplastics and nanoparticles in aquatic environments, and particle surface interactions. Professor Tufenkji's research contributions to the field have been widely recognized with numerous accolades, including the Killam Research Fellowship, the Engineers Canada Award for Support of Women in the Engineering Profession, the Chemical Institute of Canada Environment Division Research and Development DEMA Award, the YWCA Woman of Distinction Award in Science and Technology, and the Hatch Innovation Award of the Canadian Society for Chemical Engineers. She's also a fellow of the Royal Society of Canada and the Canadian Academy of Engineering. She's a professor in the Department of Chemical Engineering at McGill University, where she holds the Tier 1 Canada Research Chair in Biocolloids and Surfaces. Her academic journey includes a bachelor's in chemical engineering from McGill University, followed by an MSc and PhD degrees in chemical and environmental engineering from Yale University. And now, without further ado, it is my great honor and pleasure to turn it over to you, Professor Tufenkji.
Thank you so much, Miriam, for this kind introduction. And thank you, everyone, for the invitation to share with you some of the work that we're doing in my laboratory at McGill. Can everyone hear me okay? I hope you're hearing me all right, and that you're seeing my slides. Yes, we hear you,
and we see your slides perfectly.
Wonderful. So today, I'd like to share with you some of the work that we've been doing, focusing on detecting this small fraction of plastic particles in the environment, which is one of the big challenges when we're working on plastic pollution, is really trying to detect and identify the smallest fraction of particles. So typically when we think of plastic pollution, images such as this one come to mind. Many of us have seen pictures such as this one that depict the global crisis of plastic pollution. And when we think of plastic pollution, we often think of pollution in marine environments, such as the picture that we see here. In this next slide, I'm showing you a graphic that shows you some of the sources of these plastic pollutants specifically in marine environments. And you can see that we have many different sources of plastic pollutants. Some are more obvious, some are maybe less obvious. So for example, tire wear particles. Those are something that, you know, we are forming every day as we drive our vehicles. Tire wear is released into the environment. Marine coatings on marine vessels, but also synthetic textiles. Every time we do our laundry, we're releasing plastic fibers into aquatic environments. And so we often think about, like I said, this plastic pollution and its impact in marine environments. But in fact, what's less known is that plastic pollution is estimated to be significantly more important in terrestrial environments. And so, for example, you can think of outdoor plastics, which weather due to exposure to sun and wind. So you can think of these plastic products releasing smaller plastic fragments into the environment. Another important source that was overlooked for many years, but is now drawing a lot more attention from researchers, is plastics used in crop agriculture. So you can see here in this picture, this is a picture of plastic film, which is referred to as plastic mulch, and it's applied in different crop agricultural settings. And one of the projects that we're working on is looking at the breakdown of these plastic mulches as they're exposed to UV radiation and other forms of weathering. And so in this figure here from one of our recent papers, we're summarizing some of the different sources of this plastic pollution in crop agriculture environments. And so not only plastic mulches, but also you can think of plastic bags and containers that are used in these environments, plastic netting, plastic tubing and piping that could potentially break down, or also polymer-coated fertilizers, for example, that are applied in crop agriculture. So you can imagine, as you can imagine, in the environment, as plastic breaks down into smaller and smaller fragments, which are known as microplastics, and then particles that are even smaller, less than about 1 micrometer in size, that are called nanoplastics. As you can imagine, as these plastics break down into smaller and smaller fragments, we now have this very heterogeneous mixture of plastic pollution, of different types of plastic, different sizes of plastic particles, and different shapes. So really, it's a very complex mixture of pollutants. And because we're looking for, in our research, we're particularly interested in the very small particles, the ones that are even smaller than 1 micrometer in size, the nanoplastics. Because we're interested in these really small particles, we're essentially looking for a needle in a haystack. And the reason I say this is because we're looking for these tiny carbon-based particles in a carbon-dominant matrix. So it's extremely difficult analytically to not only detect these particles, but then to identify what kind of plastic we're dealing with. So in this figure here that I've taken from a recent Nature feature article, what they've done is they summarize at the time the different studies that were available in the scientific literature on microplastics and nanoplastics in the environment. So on the x-axis, what they're plotting is the size of particles that were studied or detected. And on the y-axis, you have the shape of those particles. So the blue circles represent the studies or number of studies that where particles were detected in the environment. What you can see is there are lots of studies and larger blue circles for the larger size of plastics on the right-hand side of the figure. And as you get to smaller and smaller sizes, as you go from right to left, you can see that you have less and less studies reporting these particles. And that's because it's so hard to find these particles or detect these particles as they get smaller and smaller in size. Now, the orange circles represent studies that were reported where scientists exposed organisms to plastic particles in the laboratory. So again, you can see, scientists have exposed organisms to different sizes of particles. You can see the orange circles kind of span the size range, but very limited shapes. So in the environment, you're going to find microplastics of many different shapes. Excuse me. But you can see that a lot of the studies, especially for the smaller size particles, were done with spherical particles. Now, I'll talk a little more about this in the next slide, but this is really limiting our understanding of how these plastic particles might impact organisms in the environment, because we really only have focused on these spherical particles. So it's not necessarily representative of what we might find in the environment. Excuse me. So we have these studies, we have this data. It's kind of limited in terms of the shape of the particles that have been studied, but also the size. You can see that on the left-hand side of the figure, for the smallest fraction of particles, there's really very little data available. Now, I mentioned to you that a lot of those studies where scientists expose organisms to plastic particles are done with these spherical particles. And this is because we can easily purchase these spherical beads from different commercial vendors. And these beads, what's nice about them is they can be purchased with a fluorescent tag. So why do we want these fluorescent tags? It's so that we can track the particles. For example, if you were going to use a water flea, Daphnia magna, that I've shown you here on the slide, and feed it these small plastic particles, well, if they're labeled with a fluorescent tag, then you can actually see where the particles are ending up inside the water flea. So here I'm just showing you how we expose these water fleas. We have different beakers or flasks, vials, and then we will fill them with a certain water and spike it with some of these small plastic particles, and then add the water fleas and do the exposure for, say, 48 hours or longer. And then after that time, we'll take the water flea and we'll look at it under the microscope. Now, the problem is that these plastic particles that are fluorescently labeled, they're available in very limited types of plastic. They're limited in terms of the size of plastic particle available. And like I said, they're mostly spherical. So they don't represent the shapes of plastics that we'll find in the environment. Another problem is that these fluorescent labels on the particles can actually leach from the surface of the plastic. So now you're dealing with a situation where you're trying to do this microscope imaging to see where the plastic particles ended up in the organism, but are you actually seeing the particle or are you seeing the free dye? You don't know because it's very hard to tell at this scale. So let me show you a picture here. This is actually a microscope picture of 100 nanometer fluorescent polystyrene beads. So these were purchased, these specific ones were not purchased labeled. They were labeled in our lab. So we purchased the beads and then we labeled them. And what you see is actually, unfortunately, we see a very fuzzy image. So we see all this red fluorescence fuzziness. We can't really tell, are these particles? Is this free dye that has leached off of the particles? So it's really hard for us to say anything conclusive from this kind of image. And that's because conventional fluorescence microscopy lacks the resolution or the ability to image plastic particles at such a scale. So what some researchers will do actually is they'll take the organism, in this case, say the water flea, and they'll digest it in a strong base. And then basically by digesting it, they get rid of all of that organic matter, the tissue of the organism, and then they'll extract the plastic particles and try to use another technique to identify the type of plastics, the size, and the number of particles. So it's actually a really big challenge for researchers who are trying to work on the small sized particles, understand where they're ending up in the environment, where they're ending up inside organisms, because the analytical techniques are just not available. So there's really a need for us to develop new methods that allow us to detect and localize where these particles are ending up inside a complex sample, such as an organism like our water flea. So in the last couple of years, my group has been working to try and develop some new methods that will allow us to detect these particles, image them inside whole organisms, and identify what kind of plastic we have inside an organism. So in the rest of my presentation today, I'd like to tell you about some of these methods that we've been working on. So the first method that I'd like to tell you about is stimulated emission depletion microscopy, or STED microscopy. And we've been using this method in particular to get around this limitation in resolution of conventional fluorescence microscopy. So in STED microscopy, what's unique here is you have your typical excitation spot here, your excitation laser, but also you're now adding a second laser, which you can see has a donut shape. And so this second laser, which has the donut shape, essentially when you use it at the same time, this donut shape is suppressing the fluorescence in the periphery of the excitation spot. So now it's essentially, you can see in this bottom image, which I'm showing you where it says overlay, that second donut shaped laser suppressing that fluorescence from the the periphery of the excitation spot. So essentially what you're doing is you're getting a sharper excitation spot. And so we wanted to see if we could use this for nanoplastics research. And of course, there are a few challenges we need to overcome. The first one is we need to find dyes that are compatible with this technique, with the STED technique. And we need to use those dyes to label different types of plastic and see if the dye is stable on the surface or inside plastic. So that's kind of the first steps to see if this is a technique that would work for environmental nanoplastics research. So when we started with this technique, we started with two different dyes, Atto 647N and iDye Poly Blue. And I'll show you some of the results that we got using this technique. So first, what we need to do, like I said, is take this STED compatible dye and get it on the surface or inside our plastic. And we need to do this for different kinds of plastics. So here we have our plastic particle. The first thing we did is just what's called a passive labeling. So essentially what we're doing is we're mixing our plastic with our dye and hope it sticks. And so here we didn't really expect this to work very well because we just thought, well, maybe the dye will wash off of the plastic, but we thought we should try it anyway since it's so easy to try. So here we mix the dye with the plastic and we hopefully get our labeled plastic particles. The next technique is actually a little more robust. We thought a covalent labeling. So taking now our dye molecule that is NHS functionalized and then covalently linking it to an amine functionalized plastic particle. So here the dye is actually covalently bonded to the plastic. So we thought this will probably work better and the dye will probably stay longer on the plastic surface. The third method that we tried is called a swell labeling approach. And so here what we're doing is we take our plastic particles, mix it with the dye, but now we're doing it in a solvent and we are heating up the mixture. And as we heat up that mixture, the plastic will expand and, expanding that polymer matrix and allowing the dye to diffuse inside that polymer matrix. We wait for a couple of hours and allowing enough dye to diffuse into the polymer matrix. We then cool down the whole suspension, and that basically allows that polymer matrix to shrink and hopefully trapping the dye inside the polymer matrix. We then wash the particles to get rid of any free dye. So the idea is that we could trap the dye inside the polymer matrix. That might be even more efficient for us. So let me show you how that worked. I don't know how well you can see, excuse me, the fluorescence imaging on your screen, but you can trust me that I tell you that there are little red dots on the screen. And what you're looking at here is STED imaging of 100 nanometer polystyrene beads that we labeled ourselves in the lab using that passive labeling approach, the covalent labeling approach, and the swell labeling approach. And you can see that the swell labeling, we did it for the two different dyes. And what's important to point out here is that you actually see individual particles. So the technique worked. We can label the plastics and well enough that we can actually detect those particles with the microscope. Now, these are very, very small particles, 100 nanometers. And what's neat is we can actually see the individual particles. And we did some tests. The dye actually stays with the plastic for up to about 50 days, depending on the solution that you put them in. And all three techniques worked quite well. So we were even surprised to see that even the passive labeling, where you're just mixing the plastic with the dye, worked quite well. So the next step is to see, okay, well, how can we use this in our research? So what I'm showing you here are those 100 nanometer polystyrene particles. They were labeled in our lab. And on the left-hand side, you're seeing an image taken with a confocal microscope. And again, you see it like I showed you before. It's a very blurry image. It's fuzzy. You can't tell whether you have particles or free dye, and you don't know where those particles are in the field of view. Now, on the right-hand side, you have the exact same field of view, exact same sample, same field of view, but now we've turned the STED mode on. And what you can see is now that you've lost that fuzziness, you really have a sharper image like we were hoping. You can see the individual particles, you can count them, but also you can see where they're localized in the field of view. So you can actually see where they're ending up inside your sample. So we were really excited to see that this actually works really well with these polystyrene particles. But of course, we want to be able to do research with different kinds of plastics, not just polystyrene. So we tested it with different plastics, and I'm showing you here some of the different plastics that we looked at. We looked at PTFE and we looked at PMMA. These are different kinds of polymers. And again, we just did the passive labeling with that dye. And I'm showing you again these side-by-side images, the confocal mode on the left and the stead mode on the right, the exact same field of view. So for example, if you're seeing a big blurry blob here in the confocal image, you can see that it's a sharper, actually, particle here in the stead view. And again, you can really see it with the PMMA, where it's a very, very fuzzy, blurry image in the confocal mode, but in the stead mode, you can now see where you have these individual, very small particles. So we were happy, we had success with different kinds of plastics, but we wanted to see, can this be used to find and detect and localize plastic particles inside a whole organism? Much more challenging scenario because what I'm showing you here is just in water. So now let's look at what happened when we fed our worms some of these plastic particles. These are now 50 nanometer plastic particles, very, very small. And what I'm showing you on the far left is an overview image of the roundworm's mouth. And the little white square, the dash square, is where we're going to zoom in. So the central microscope image here in confocal mode is that zoom in from panel A. So we're looking at this little zoom in the white square. And you can see quite a blurry image. You can't really tell where those particles are. But when we turn on the stead mode, you can see those individual particles. You can see where they ended up, and you can zoom in even closer. So this is, for us, it was progress. We can now see these really small particles inside a whole organism. We can achieve this single particle resolution, but there are still limitations because we need to use particles that are labeled with this fluorescent dye. It has to be a stead compatible dye. And this technique does not tell us what kind of plastic is in the organism. In this case, we know because we fed the organism polystyrene, but if we were to take an organism from the environment, we would not know what kind of plastics there were there, and we wouldn't necessarily be able to find them because they wouldn't necessarily be stained with this fluorescent dye. So it's a good technique to use in the laboratory for our controlled studies, but practically speaking, it's not going to be useful for us to study samples from the environment, because in the environment, we're going to collect potentially organisms that are going to have ingested plastics that are not labeled. So in this second part of my presentation, I'd like to tell you about a technique that we have worked on where we were trying to image small plastic particles inside an organism, but those particles were not labeled before being ingested. So let me walk you through what we did here. I'm showing you here a water flea just as a representative organism. We took our water fleas and we fed them microplastics. Those microplastics were not fluorescently labeled or labeled in any way. So the organism takes up the microplastics, and you can see here the gut is filled with microplastics. What we did next is we took that whole organism and we soak it in a fluorescing dye solution. So what happens there is now the dye is staining everything. It's staining the particles inside the gut, and it's staining all of the organism's tissue. So you might be asking yourself, why are you doing this now? Why are you staining the whole organism? Now you've got a big problem because everything is fluorescing. So what we did is we took this organism that is all dyed, the tissue and the plastics, and we soak it in what's called a tissue clearing solution. And the idea here is that this tissue clearing solution actually loosens, if you will, the tissue, and therefore releasing the dye from the tissue. And the hypothesis and the hope is that by releasing the dye from the tissue, you now have an organism that is cleared, it's more transparent, but you still have, excuse me, a gut filled with microplastics that still have dye attached or associated with them. So the idea is that by soaking in this tissue clearing solution, we won't impact the microplastics that have been dyed, but we will kind of clear and make transparent the organism itself. So it's actually easier to look at under the microscope and to see these microplastics that have been dyed. So let's take a look and see if that worked for us. So here first, I want to show you what happens when you soak these organisms in this tissue clearing solution. So we have here three different organisms: our water flea on the left, Tigriopus californicus, which is a shrimp-like organism, and then the cephalochordata, which is more like a fishy organism. And on the top, what I'm showing you are those three organisms, which are before they've been soaked in a clearing solution. So you can see they're not so transparent. I mean, the Daphnia is somewhat transparent naturally, but the other two are not. And you can see the cephalochordata, for example, you cannot see the grid lines through the tissue of the organism. On the bottom set of images, what you're seeing are those organisms after they've been soaked in a tissue clearing solution. And you can see what happens is that these organisms become more transparent, and we can see the grid lines now through the organism tissue. Especially you can see it with the cephalochordata. Now you can see those grid lines through the organism's tissue. So we've figured out, okay, how long we need to soak these organisms in a tissue clearing solution to be able to make them more transparent. That's the first step. Now, the next step is to understand, well, if we have these plastics and they're dyed and we put them in a tissue clearing solution, will the dye wash off the plastics? So we had to do some tests to verify this. So what we did is we took different kinds of plastics, soaked them in a dye, and then soaked them in a clearing solution for up to 28 days, and then looked at their fluorescence again. So here what I'm showing you in the top set of images, these are 6 different plastics that we looked at, and what you're seeing is their fluorescence at day 0. So they've been dyed and soaked in a tissue clearing solution, and right away at day 0 we look at their fluorescence. And then you can see their fluorescence in the bottom set of images after 28 days. And what you see if you compare the top and bottom image for each plastic is that the fluorescence intensity in these images is relatively constant, so it's stable. So you're not losing a lot of fluorescence intensity over 28 days in a tissue clearing solution. And that tells us that the dye is actually staying on the surface of the plastics, which we were very excited about. So now we can dye plastics and then soak them in this tissue clearing solution up to 28 days, and the plastics are still fluorescing. So the next step is to see, well, does this actually work in a whole organism? Does the whole scenario work? So to remind you, we take an organism, we feed it plastics, they're not dyed. We then soak them in a dye, the whole organism, dye everything, and then soak it in a tissue clearing solution, hoping to release the dye from only the tissue. And let's see if it worked. So here we have our water flea, and this is what it looks like under control conditions. So, you know, no plastics exposed, no, I actually here, yes, it was, I believe, dyed and cleared. And then on the right-hand side, you have the water flea that's been fed microplastics, 10 micrometer microplastics, then dyed, and then tissue cleared, and an image. And what you see is that the gut of the water flea is filled with these pink particles, which they look pink at this scale. And so we can really see that those particles have ended up in the gut, and we're not really seeing that pink fluorescence anywhere else in the tissue of the organism. So the technique really worked to release the dye from the tissue of the organism. And also with the shrimp at the bottom, shrimp-like organism, we can see a similar kind of result. So we have the control where you don't really see those spots of accumulation of particles. And then on the right-hand side, you see the accumulation of particles, different parts of the organism. So theoretically, this technique could be used to look at microplastics in organisms that we collect from the environment. And that's because we developed this technique to look at microplastics that are not dyed before the organism ingests them. All of the dyeing was done after the organism had ingested these plastic particles. So the next step is to try and use this with organisms that we've collected from the environment. Let me show you another example of this technique. at one point during COVID, actually, I kept finding these masks all over my neighborhood, in parks, in parking lots. You see them everywhere, just people throwing them out indiscriminately. And so my question that I asked myself is, well, these masks, they're made of plastic materials. Are they releasing microplastics, nanoplastics into the environment as we walk over them, the cars run over them? And they're left to be weathered by the environmental stressors. And so we took some of these masks and we kind of weathered them in the lab, and we collected the particles that were released from these masks. And then what we did is we exposed some different Daphnia, these water fleas, to these particles that had been released from the masks. Now, of course, these particles released from the masks, they're not fluorescent. So we exposed our water fleas to these particles, and then they ingested the particles, and then we ran them through our procedure, which was to dye the whole organism. So this dyes the particles, and then tissue clear, and then image. And here we're seeing the results. So you have the neonates, which are like the little babies, if you will, and then the juveniles and the adults. And you can see that as they grew older, they were more likely to take up more and more particles. And we were able to really see where the particles are ending up inside this organism. So the technique does work to allow us to detect plastics inside a whole organism. But what it doesn't allow is for us to actually detect or identify what kind of plastic we have there. So this technique is good for imaging, for localizing, but not telling us what kind of plastic we have inside the whole organism. So in this last part of my presentation, I want to tell you about a new technique that we've been working with that will allow us hopefully to detect and identify what kind of plastics we have in our organisms or other kinds of environmental samples. So the technique is called optical photothermal infrared microscopy, or OPTIR. And here, the technique does not require us to have labeled particles. It's a non-destructive technique. And what you're doing is actually IR and Raman mapping simultaneously. So you get both IR and Raman spectra of our particles. Now, very different from conventional IR microscopy, with OPTIR, we're actually monitoring the change in the photothermal response of the material as it absorbs IR light. So as the sample absorbs IR light, it expands, and it's that thermal expansion that we're monitoring using a visible light. And because of the wavelength of that visible light, we can actually access or achieve much lower resolution. So we can achieve roughly about 0.5 micron resolution with this microscope. So we can go to much smaller particle size than conventional IR microscopy. So let me show you a little bit what we've done with this technique. So just starting out, these are just really simple spectra that we've acquired of different plastics. So we have polystyrene, polypropylene, polyethylene, and PMMA. And what I'm showing you in red are the IR spectra, and then in green, the Raman spectra for these plastics. And this is really simple. The plastic particles are just suspended in water, as you can see in the bottom bright field images. So what I'm showing you is just the average spectra from 5 different particles that were selected kind of randomly. So you can see we can get that IR spectra, but also simultaneously the Raman spectra. Now, this is, of course, very simple to do. We have 4 individual types of plastics in water. It's clean, it's easy. But of course, our research requires us to work with more complex samples. So here I am showing you an image of taken using plastic mixture. So here we've taken our 4 different plastic particles and mixed them, and see if we can identify the different plastics in the mixture. And so what we've done here, it's a little different from what I showed you in the previous slide. Here we're actually mapping at selected wave numbers that are characteristic for each plastic. So for polystyrene, for example, we're mapping at 1601, for polypropylene 1378. So we're really, and this is just to save time, because it would take a lot of time if we were to map using the entire spectrum. So you can see in these images that you can now identify where we have our different plastics in the sample. And we can merge those images to see how these different plastics, where they're located vis-a-vis each other. So we can look at mixtures, but again, this is just really clean water, so it's relatively simple to do. Now, of course, in our research, what we really want to do is to be able to identify and locate these plastic particles in more complex samples. And imaging and identifying plastics in something like an organism, like I said, is a big challenge. And especially with IR, the challenge here is that the biomass actually interferes with your IR imaging. And so here I'm showing you a representative spectra of biomass, and you can see the proteins have a strong signal and the lipids have a strong signal. So to try and get around this, what we decided to do is see if we could actually cut our organism into very thin sections. And by doing that, we could potentially minimize this interference of the biomass. So essentially, we took our organisms that have been exposed to plastic. Again, these are not labeled plastic particles. They're just regular plastic particles. Organism takes up the plastic, and then we take it to the microtome, and the microtome slices our organism into thin slices, which are placed on glass slides. So each slice is placed on a separate glass slide. We can then take those glass slides to the microscope, this OPTIR microscope, and take a look at it. And hopefully, because we have, really, we're looking at a very thin slice, hopefully we've reduced or minimized the impact of the biomass, the tissue, in our IR imaging. So let me show you what we did here. First, here, this is with the water flea, and we fed it separately, either polypropylene, polyethylene, PMMA, or polystyrene. And you can see the polystyrene is actually really small. It's submicron at 750 nanometers. And all these exposures are done with separate types of plastics at 1 ppm. And then what we did is we sliced them up, and here we're looking at the slices around the gut. And we're pointing with the arrows at where we took our spectra. And what I'm showing you in the bottom are the averaged spectra from those particles. And we've identified with these red or pink bands the characteristic peaks for each of those plastics. So you can identify, we were successful here, we can identify the different kinds of plastics inside the whole organism after it's been sliced. So this was good. This was quite successful. In our lab, we're also interested, of course, in terrestrial organisms, because terrestrial plastic pollution is also of interest. And so we work with Drosophila melanogaster, or the common fruit fly. Here we did the same thing. We fed our fruit flies different kinds of plastics, and then we took it to the microtome, sliced it up, and here we're looking in the gut region, and we can identify the particles visually because we fed them spherical particles. And this was done to develop the proof of concept, to make sure that what we are actually imaging is what we think we're imaging. And in fact, that facilitated the development of the method. So here again, we've confirmed using those characteristic bands that we have polypropylene, polyethylene, PMMA, and polystyrene. So this was a good first step for us, that we were able to develop this method and show that it works for these individual plastics. But of course, in the environment, our fruit flies, our water fleas, other organisms are going to feed on a mixture of plastics. So we wanted to see, what can we do if we feed our water flea, for example, a mixture of these 4 plastics? And they were at a concentration of 1 ppm for each plastic type. So here what I'm showing you is actually the slice right through the water flea. And you can see the head at the top, the front here, and then the gut is very clearly outlined. And this little red square here is what I'm showing you in the bright field image and in the OPTIR map. So what we did here is a little different from the previous slide. Here, instead of doing this full spectral analysis, we are choosing a characteristic wave number for each plastic. So for PMMA, it's 1725. And for polypropylene or polyethylene, it was challenging to differentiate between the two, but we worked with 1465. And what you can see is you can actually now localize inside that slice where you have different kinds of plastic. We had a tough time finding the polystyrene inside this slice because, again, it's so small, it's smaller than 1 micrometer. We did the same thing with the fruit fly. So now here again, you can see the head of the fruit fly, the wing. It's a slice right through our fruit fly, the abdomen. And then here, this red square is what we're looking at. And we can see these particles here. We've mapped them with the OPTIR. And again, you can see where you have some PMMA. It's also taken up some polypropylene and or polyethylene. But again, we had trouble seeing that polystyrene. So in order to see the polystyrene, we decided to go back and try this, but by doing this targeted spectral analysis. So now if we look at that fruit fly again and we look at a slice, and we now select some spots that look like they have spherical particles, and we do what's called this full spectral analysis on those particles. This is what we get as the red curve. So we have that red curve, we can see those ingested particles, and we compare that to bare polystyrene or just a spectral analysis of biomass. And we can see, well, this red curve actually shows us characteristic bands for polystyrene. There's also some interference from the biomass, but we can be pretty confident from the peaks that we see that we do have polystyrene there. So we have to tweak the method sometimes depending on, the size of the particles. There might be some different kind of analysis required. But we were happy to see that we can even detect and confirm the identity of particles as small as 750 nanometers. And finally, we were working with some colleagues at the National Research Council of Canada, and they were interested in earthworms, which are another example of an interesting terrestrial organism. So we took our earthworms that we had fed, in this case polyethylene, and if we zoom in here to this region of the earthworm, you can see these spherical particles. And if we do this targeted spectral analysis directly on those spots, we can get the characteristic peaks for polyethylene. So we were successful in being able to localize, detect, and identify different kinds of plastics in different kinds of organisms using this approach. So to summarize, I showed you in the first part of my presentation how STED microscopy can be useful for laboratory studies where we want to localize single nanoplastics inside complex samples. In the second part of my talk, I showed you how we can use that tissue clearing approach to localize microplastics, even if they were unlabeled before uptake by the organism. And in the third part of my presentation, I shared with you some of the work we've been doing using OPTIR combined with histology techniques to detect, localize, and identify unlabeled plastics in a whole organism. And our next step in our research is to analyze samples that we collect from the environment and push the technique to more and more complex samples. So before I finish, I want to acknowledge all the wonderful students and postdocs and collaborators that I worked with to achieve this work. Junrei McCarran, who was a postdoc in the lab, and Frank Lee, who was a master's student. Arav was also a master's student while we were doing this work, and Brian was a postdoc in my laboratory. And then we worked with our collaborators at the NRC and at McGill. Want to acknowledge the funding agencies. And thank you again for the opportunity to share with you some of the work we've been doing at McGill, and I welcome any questions that you might have.
Thank you so much. That was really fascinating. And I really appreciate how you made us all feel like we were right there in the lab with you, kind of discovering things. So it was really, really fascinating and timely research. And I think particularly timely because right now, over 180 countries are meeting in Geneva for renegotiating or trying to come to find a legally binding global treaty on plastic pollution. So I guess with that context, I want to ask you, how can cutting edge research like yours help inform what's included in the treaty, especially with detection, monitoring, and life cycle accountability? What do you think of the role for technologies like STED or OPTIR in future treaty enforcement?
Thank you so much, Miriam. First of all, for the invitation and for this interesting question. I think all the research that's being done, clearly not just ours, but all the research that's been done on developing techniques to advance our ability to quantify and characterize plastic pollution at all scales is so important in terms of feeding information and real data required to make these kinds of decisions and develop policies. You know, without right now, like I showed you, we have very little knowledge of nanoplastic or microplastic pollution loads in the environment. There's even more limited knowledge in terrestrial environments, you know, but science is advancing very fast. You know, I just came back from a Goldschmidt conference in Europe, and you know, I heard lots of interesting presentations on new techniques being developed to quantify and characterize nanoplastics in air samples, but that can also be used for water and soil samples. So all that to say, I think, you know, the science is advancing. We need to see more implementation and more environmentally relevant conditions and environmental samples. And all of that data is going to help support any kind of policy development, but also, like you said, implementation of those policies and enforcement of those policies.
Thank you. Yeah, I appreciate the work, the thoughts, you know, that have gone into all this is great. But right now, I don't feel like a scientist. You know, I just want to talk like a human being. You know, I saw a documentary not long ago about the level of plastics that gets into our food from the kitchen cutting board. You know, so right now we can't use plastic cutting board in the kitchen. Another documentary says you can't even use wooden cutting board, you know, to cut your meat or your chicken in the kitchen. And I think at some point when the use of plastics was discovered as packaging, the whole world was jubilating, oh, we've seen something that is waterproof, that we can use. A lot of money went into that, you know, across all sectors. And all of a sudden, scientists are now saying, no, plastics, micro, nanoplastics, they're now an issue. My question right now, as a lay woman who cannot use either plastics or wood to cut our food in the kitchen, what should we do differently? What should we use? You know, should our attention be to finding very quickly now the solution, you know, or the alternatives to plastics, you know, or what? I'm just confused, a lay woman. Thank you.
That's a great question. Thank you so much. You know, I think it's a very complex question. So one of the things that I guess one part of my answer is related to, you know, what you said about we don't know what to do. And we also, there's a lot in the news about microplastics and how much we're ingesting. I think as scientists, we have to be very careful to inform the public without being alarmists. So, you know, clearly it's in our food, it's in our air, it's in our water. We can't get around it. We can't avoid it. I think we're going to have to reduce, you know, the use of plastics to reduce introduction into our environment, exposure. We also have to develop more remediation techniques to try and reduce exposure in that respect. So that's part of my answer. The other part of my answer addresses your question, you know, what do I do, right? To be honest, I'm still using both the wooden and plastic cutting board in my own kitchen. I think right now we don't have a lot of options presented to us. And until manufacturers start developing alternatives for us, we're going to be limited in our choices. And, you know, I think it's up to us to push for those options, to push for those alternatives, and to put pressure for the development of materials that are less toxic. Just like we're seeing now with a lot more pressure being placed on the development of materials that include less PFAS, for example. So I think that's where, you know, we're moving in the right direction in terms of more information, more awareness. But I really value your question. It's a really, really important question. What do we do? And you know, I think we do the best with what we have at this time and hope that we can inform and educate so that we can develop better and better approaches and better lifestyle options for everyone. I hope that addresses partly at least your concern or your question.
Thank you, my dear, for your presentations. Very useful and informative. My question, I'm wondering if, in your opinion, whether this technique can be used for and detect microplastic in wastewater, and if it's can be useful to track parasites in wastewater, and considering the selectivity of the daily use, whether there might be interference and false positive images or reasons.
Thank you for the question. I'm sorry, I didn't hear most of the question. Is it possible to repeat it or type it in the chat?
Okay, I'll try to repeat my question. I was wondering if your technology can be used in wastewater. I mean, in detection of nanoplastics or tracking parasites, for example, within wastewater effluents. I'm also wondering about the selectivity of the day use, the stain, if there is any kind of interference, false positive, that can affect the results. Thank you.
Thank you so much for the question. So the first part of the question, the technique should be able to detect plastic particles in wastewater samples. The sample would have to be filtered so that the sample is on some kind of filter or slide that we can image in the microscope. For the second part of your question, well, regarding parasites, I'm not so sure. I haven't worked with parasites with these techniques, but I suppose if they have some kind of characteristic spectral signature, then that could be a possibility. And regarding the detection of false positives or the specificity of the technique, so with the IR technique, it's really quite specific. You can really characterize what kind of material you have there, and you should be able to differentiate between, as we saw, different kinds of plastics and different non-plastic materials. We've been doing some work with other kinds of environmental samples, and our preliminary data show that we can actually discriminate between different kinds of materials. With the other techniques that are dye focused, there is the risk, of course, of dyeing something that is not plastic and then detecting it. It's not something that we've really worked on, because here we were really trying to develop a method for plastics, but it is something that we can definitely explore to try and understand what kind of other materials these dyes might interact with.
Thank you so much for the presentation. Mine is a clarification. I want to know if the water filtration process is able to detect and remove microplastics from water that we drink, like tap water.
So I think if I heard the question correctly, is it that our conventional water treatment processes, whether they remove microplastics?
Yes.
So thank you for the question. Actually, so I mean, conventional water treatment processes are designed to remove particles in general, so they will remove a certain proportion of plastics. What we found in our laboratory tests is it's much more challenging to remove the nanoplastic fraction. So we're going to need more advanced techniques or approaches to deal with the nanoplastic fraction. And that's something that we've been working on in my laboratory. I didn't talk about it today, but a certain fraction of my laboratory is focusing on techniques to deal with or advance our ability to remove the nanoplastic fraction. And, we've done some work on different kinds of plastics. So microfibers versus beads versus tire wear particles, and then of course the nanofraction. So all of this research is starting to be done, especially with the nano in different laboratories. And it's starting to point to the fact that right now the approaches we use don't necessarily deal with the nanoplastic fraction very well.
Thank you.
Thank you.
Yeah, thank you. Thank you. Very nice, I think, fascinating presentation. You really well we are actually listening to like what Miriam said, it is science experiment happening in front. My question is related to the current negotiation happening to develop policy around the world about plastics. So what should we aim for those legally binding policy to aim for? What should, is it limiting, because we cannot live without nanoplastic, even with microplastics, because nanoplastic is even more finer particle. So where should we aim for those kind of policy that we are looking for legally binding? So what is your suggestion about those policymakers?
Thank you for the question. So personally, my personal opinion is that we need a very balanced approach. So we're going to need to have policies that impose reduction of production and reduction of use, but also policies that mitigate or impose improvement and increase in our recycling, our collection, our reuse. So, I think we need to deal with the problem on both ends. Not just the source, but also where these plastics are ending up. I really truly believe in our ability to deal with this problem, with the right willpower. We do have the ability as a global community to deal with this. It's just a question of will. And, we have to look at it really as a global problem, not just, one aspect of it.
Thank you.
Okay, I researched the energy system. Below, there are a lot of wind power and solar power in the world. I want to know when the clean power have retail, how to deal with this clean machine.
Thank you for the question. I'm sorry, I didn't completely understand the question.
I researched the energy system. Below, there are a lot of wind power and solar power in the world. I want to know when the clean power have retail. So how to solve this clean machine?
Do you mean
I'm
to how to
sorry.
clean how to clean the
Yeah.
solar panels and
Yeah.
in a way that doesn't
Yes.
doesn't release microplastics?
Yes.
Okay. Chung is a visiting professor whose research is on renewable energy, and he's asking about the growing increase in solar power and wind power. What are technologies that we can use to safely clean these large-scale renewable systems without releasing microplastics, either from the systems themselves or in the cleaner?
Well, it's an interesting question that I've never really thought about. I don't really have an answer to your question. It's not something that I'm familiar with. I'm sorry.
Well, thank you so much. Unfortunately, we're out of time now. I think we could all continue talking to you for a long time. Really, really appreciate your taking the time to share your very exciting research with us. Congratulations on this really important work. And I was looking at your lab, and it's amazing just the diversity of different areas that your graduates of your lab have gone on to. So I think especially in the context of these negotiations, that really gives me a lot of hope. Thank you very much for taking the time to share this with us. And we look forward to seeing you all again next week.
Thank you so much. Thank you, everyone. Thank you.